Okay, guys, in this lecture we shall be talking about DNA recombination and the various techniques used for the DNA replications. Okay, so let's talk about DNA replication. Well, okay, so DNA recombination, as we know, it is a technique which is basically used for you know like combining two DNAs and getting a new one. Well, that is basically a hybridization of it, but somewhat like that. And DNA cloning is just another method which is used to you know like DNA cloning is a method which is used to clone, like make the replicas of the obtained genes, and they are cloned like they are made in many copies. So that is basically DNA cloning. And the recombination technique is basically used to mix the other genes or the other site of interest in the DNA, and they are combined together to obtain a new one. So that is somewhat called as recombination. So the construction of a recombinant DNA molecules that can be made by this technique, which is autonomously replicating DNA segments. So if we get our gene of interest, so what we need is just the autonomously replicating DNA segment that is in the whole DNA molecule. All we need is that one segment or that one gene which we are interested in. We are interested in sorry okay which we are interested in, and the inserted elements. It is basically the segment which needs to be inserted into it, and the combination of this both is known as chimera. So recombinant DNA molecules are made by attachment of the DNA fragments in vitro to the target DNA to a replicon. So replicon here is a particle, sorry, it is a DNA site which can replicate by its own, and that is replicon. Which can replicate is called as a replicon. So replicant is any sequence which is independently replicating. Okay, so the methods which are used for the recombination of DNA are known as transformation. So transformation is basically the transfer of the genetic material from one bacteria to the other, and this is known as transformation. So the second is selective propagation of the cell clones. Oh, sorry, I forgot over here. There are two types of cell cloning, which is in vitro. Sorry, there are two types of cell cloning: in vitro and cell based and cell free. Okay, so in cell based technique, what happens is the DNA cloning is of two types, which is cell based and cell cloning. In cell based, basically the whole process is carried out in cell, and in vitro and cell free, the whole conditions are carried out in lab conditions and not in cell conditions. So this thing which we are discussing, it is about the cell cloning, cell cloning, which is cell based. Okay, so transformation that happens between cell to cell contact. Okay, and this how does this transformation takes place and everything will be explained in further in future lectures. Okay, so selective propagation of the cell clones. So the transformed cells are plated out, means basically the cells which are which have which in which the genetic exchange takes place. So these cells are basically transformed cells. Okay, so as the genetic material exchange takes place, the cells obtained are the transformed cells, and these transformed cells are then allowed to grow on a on the general media, and then the individual colonies are picked, and the cells can be further explained expanded in liquid culture. So basically they are allowed to grow in a normal agar medium, which is the nutrient agar or any specifications if we need. And this are and then the individual colonies are picked up and they are allowed to grow on a liquid culture. So next is isolation of DNA clones. So the isolation of the recombinant DNA clones by harvesting cell culture and selectively isolating the recombinant DNA. So basically the DNA is basically the DNA is the recombinant DNA is isolated, and the clones the clones are basically harvested and they are selectively and they're isolated, and the obtained is called the recombinant DNA. So that's it for this today's lecture, and I'll be posting now videos on a regular basis, so I hope you will be tuned in, and please subscribe. Thank you.